Thank you for the kind introduction. And it really is wonderful to be back. I'll thank the members of the search committee for inviting me to come talk to you. I had great meetings with faculty today and with the students. I saw a lot of wonderful things. I think there's a wonderful positive energy here that you should be proud of, and so many interdisciplinary fields of research coming together under the guise of neuroscience. It's very nice to see. One of the most surprising things that I heard actually came from my student driver who drove me from the airport. I was, talking about, being at UMass, and finally he said, "Oh, and when did you graduate anyway?" And I said, "Oh, 1992." And he said, "Oh, that was before I was born." Anyway, so today I wanted to take the opportunity, obviously, to tell you the work that that my lab has been doing. I'm going to paint with relatively broad strokes because I want to give you just an idea of the kinds of questions that we are asking and the kinds of methods that we're using to address them. And so I'll start off with that. I'll talk about some published work, a little bit of unpublished work, and then I'll talk about some future directions and where I see my lab going in the next 5 or 10 years. So obviously, a fundamental question in neurobiology is how neural circuits or even individual neurons contribute to behavior or perhaps or sensory perceptions. And of course, a lot of people are working on this question in a number of different ways. My laboratory is interested in looking at these questions in the olfactory system. And the reason that we use the olfactory system is that for many reasons. I think that the olfactory system, as I hope to convince you in my talk, is an extremely tractable system in a number of realms. So genetically, it's very tractable. It's quite accessible to manipulation, physical manipulation, also to optical imaging and electrophysiology. As you know, a lot of people who are working in neurobiology are beginning to use mice if they're working on mammals. And the reason that a lot of people are doing it is because these animals are quite genetically tractable. The next thing that people often learn about mice is that they're extremely olfactory. And so if you want to work on behavior, and you want to get the animals to learn to do particular behaviors, they're actually very good at picking up olfactory tasks, right? So we're working on the mouse. Mice, of course, will use their olfactory systems to do a number of things, right? They use them for mate recognition, for evaluating food sources, and for, to avoid predation. So I said that the mouse olfactory system is relatively accessible. Here it is, right? So here's the mouse. Here are the olfactory bulbs, which sit between the eyes, making them very accessible. You simply can thin the bone over the olfactory bulbs and look at least at the dorsal olfactory bulbs. Here's the nasal cavity. If you take the structure and you turn it sideways and you cut it sagittally, it's something like this. And this is a whole mount of from an animal, a transgenic animal or a knock-in animal, in which sensory neurons that express a particular odorant receptor gene are made to turn blue because they express TALAC-Z. And so what you're looking at here is the medial aspect of the olfactory bulb of the animal. This is the nasal cavity. Here are the turbinates. And you can see these individual blue dots are individual sensory neurons that have turned themselves blue because they're expressing a receptor called M71. And you can see that the axons of these sensory neurons penetrate through the cribriform plate of the ethmoid bone, and they project and ultimately converge onto a point called the glomerulus. Each of these sensory neurons is expressing, most likely, a single receptor gene. And so what you have in the animal is this convergence of what we now know are functionally similar afferents. And there are actually 2 glomeruli per bulb per receptor, right? So 4 glomeruli on average for each of the receptors. And of course, there are many receptors. This is a phylogenetic tree of all mouse odorant receptor genes, clustered by similarity from their amino acid sequences. And so it's quite a bristly tree. There are over 1,000 members. These are classical class A GPCRs, like beta adrenergic receptor or rhodopsin. And there are a couple of hallmarks of these receptors that are similar to other receptors as well, which is that these receptors can bind multiple ligands. And what we also know is that different receptors can bind to the same ligand. And I'll show you what the consequence of that is in a second, right? And so we have lots of receptors. This is the images of the olfactory bulb from a different animal in which all glomeruli are made to turn green because they're expressing, actually in this case, an activity-dependent GFP. But you can see now that the outer layers of the olfactory bulb are absolutely chock full of these glomeruli which cover the surface, the input layers on the surface. And genetically, we can do a number of things. We can genetically tag particular glomeruli. Here's a particular glomerulus, actually M72, a different gene shown in red. So the idea is that genetically we can address these specific inputs, and there are a lot of them. And this organization, and work even prior to this organization being known, emphasized that the system is really a parallel distributed processor, right? You have olfactory coming in through 1,000 parallel inputs that are functionally, quote-unquote, redundant, and that somehow the system uses the cross-fiber pattern, uses the activity across these inputs to encode the identity of a given odor and a given molecular structure. So what does that look like? So here are images from these animals, essentially, right? Animals that are expressing an activity-dependent reporter. And what you can see is that when you put on, here in this case, 3 different chemicals, 3 different odorants, you activate multiple glomeruli. Different odorants will activate different subsets of glomeruli. And what you'll even see as I go on is that different classes of chemicals, in particular the amines, activate preferentially particular glomeruli in a given region of the dorsal olfactory bulb. So these data, and of course data from decades of research, have given rise to the idea that coding in the system is combinatorial. If you look at how the glomeruli respond with an increase in concentration, what happens is that as you increase concentration, you recruit receptors, you increase the number of glomeruli that are activated. And so there are a number of interesting questions that arise from these kinds of activity patterns. For example, if you were to compare, so I've shown you that different odorants give you different patterns of activation. This is the same odorant, in this case hexanone, at different concentrations. If I were to show you this pattern and this pattern, how could you tell that that was 2 different concentrations of the same odorant or 2 different odorants? So there are a number of interesting questions that arise from this kind of a pattern of activity. I'm going to touch on these sort of tangentially. The main question that's driven the work in my lab and with my collaborators in the past few years has been this question of functional redundancy in the system. So the question is, how much information is contributed by each of these individual inputs in a system, in this parallel distributed system? So we're all familiar with the textbook example of trichromats, where you have 3 different colored opsons, and mutations or alertism at the red-green locus will give you different forms of color blindness. And so here we have 3 different input channels, and if you modify one of them, you can get certainly a very strong change in color perception. In the olfactory system, it's been much more difficult to show similar effects and to understand what the contribution of each of these individual receptors would be to olfactory performance, because we have many more receptors. And unlike in the visual system, we don't really know what to put on the axis here, right? Chemical information is much harder to characterize in some linear axis, such as wavelength. And this raises some very basic questions. So, I was trained in a classical olfactory lab, and the idea was that odor representations are highly combinatorial, and if you were to remove an individual gene or an individual glomerulus from an odor representation, you wouldn't have much of an effect on odor perception. From a systems neuroscience point of view, that makes perfect sense. From a geneticist point of view, it's a little bit of a problem, because if that's true, how is it that animals, like a mouse, how is it that animals retain large gene families over evolutionary time. If you remove one of the genes that doesn't have a big effect on behavior, how is it that they maintain such large families? Why isn't that receptor already gone? And you can make arguments about, over evolutionary time, small amounts of selection over long periods of time can help maintain this family. You can also have gene conversion and things like that. But it raises very fundamental questions about what determines the size of the olfactory receptor repertoire in a mammal, and how much each of these inputs contributes to behavior. So there are a lot of challenges in studying olfaction, which I think you can appreciate now from what I've said. We have many, many odorants. In fact, it's hard to even define how many odorants are out there in the world, but there's lots of them. There are a lot of them, and there are a lot of receptors. And so pairing receptors with odorous ligands is very difficult. That's made more difficult by the fact that functional expression of these GPCRs in vitro has met with limited success. In fact, when you do find ligands in vitro and you go to use them in vivo, they often don't work. And so it's actually been very difficult to work on the system for these reasons. And so as a result, if as a neurophysiologist you want to be able to put a chemical on the animal and have it breathe in the chemical, and you want to be recording from neurons that you know are going to be activated by that chemical. And that's been very challenging, a very simple thing, right, for a sensory biologist. You want to be able to stimulate the cells that you're recording from, and that's been somewhat of a challenge, particularly when using responsible chemical concentrations. So with all of these problems, you might imagine, well, why would you even do this, right? Well, what I hope to convince you today is that some of the progress that my lab has made and that some other labs have made finally are giving us hooks, genetic hooks that we can use to begin to study the system. And what I think that we need, to a certain degree, is listed here. What I said is that we really would like to identify ligands that activate defined receptors at physiologically relevant concentrations in vivo. And in addition, we would like to identify glomerular inputs that, if we were to manipulate them, or manipulate them somehow, that you could actually affect odor perception in some way. And we're just now getting to a place where we can begin to do this. And that allows us to begin to address some of these basic questions that I've talked about, right? So we want to be able to manipulate olfactory inputs in particular ways, and to measure what the neurons associated with that glomerulus are doing, and also what the animal is doing with that information. And so we've taken two general approaches to do this, and this is sort of a simple-minded approach here. So the first way to do this would be if you want to ask how much each glomerulus, or each input, contributes to olfactory function, you can try to stimulate individual glomeruli and see how that affects the animal's performance in an odor-guided task. The other thing that you could do is you could go in and genetically remove particular glomeruli and see what effect that has. So I'll talk very briefly about this first approach. We have two papers published on this method. The idea is that, like many people, we are using optogenetics. And optogenetics in the system allows us to address this glomerulus using light in a way that's just not possible using odors, because when you put on even very simple odors, you will activate typically multiple glomeruli. And in fact, defining how many glomeruli and how many receptors you've activated is quite challenging, because the methods that we have for doing that using optical imaging are not particularly sensitive. They're certainly not as sensitive as the animals are. Is this a trivial question? Well, not so much, because there are data out there suggesting that activation of single input channels in the system may not be sufficient to get neurons in higher cortical centers to be activated. This is a paper from Ian Davison when he was working with Mike Ehlers. And what he showed was that if you activate the glomeruli artificially using light, uncaging glutamate, what he found, and then recording from neurons in the piriform cortex, what he found is that if you put activity patterns on the bulb that were sparse, he was not able to activate cortical neurons. If you were recording from the same neuron, if you activated multiple glomeruli in a distributed pattern, you were much likely to get one of the downstream neurons. And so the idea was that perhaps the system is tuned to respond to patterns like this, but not so much sparse inputs. That obviously raises some important questions about how the system works. To address this, what we did was we used a gene targeting strategy to express channel rhodopsin under the control of the promoters from one of these receptors. So this is a cartoon of an odorant receptor gene. The good thing about odorant receptor genes is that they're very simple. Typically, they have one upstream exon. Right, this is a linear piece of DNA. The exons are shown as thick boxes. They have typically one upstream exon. The coding sequence for the receptor is intronless, usually. So the coding sequence is in one contiguous bit of DNA. The genes are relatively compact and easy to modify, and this tends to work relatively well. What we do is we use homologous recombination in ES cells to modify this gene to put this sequence after the coding sequence for the receptor. And this sequence contains an internal ribosome entry site, as well as anything that you want. In this case, channel rhodopsin, which is a light-gated cation channel fused to yellow fluorescent protein. And ultimately what this does is it takes cells that would normally choose to express the M72 receptor, and it fools them into also expressing channel rhodopsin YFP. And so what you end up with is a set of glomeruli that are now yellow because they're expressing channel rhodopsin YFP. They're also expressing their native receptors. And now this gives us a light addressable glomerulus. We can put a light cannula on top of the animal's head, and we can then use light to stimulate that particular input. And so animals are head fixed, and we direct a laser through an optical fiber over the glomerulus. In this experimental setup, we're also measuring sniffing. For the experiments that I'm going to show you, that's sort of irrelevant. We can then present odors to the animal in a controlled way using a computer-controlled olfactometer. And in this experiment, the animals are doing a go, no-go task. We can ask them, if you, for example, if you smell something, lick. If you don't smell something, don't lick, withhold licking. So in that way, we can ask them what they're smelling. If you ask an animal to do an olfactory task and you look at the performance in this go, no-go task, when the stimulus is an odorant, they obviously will do this relatively well after their training, so they're up at 90%. If one session after that you switch them to light in place of odor, what you find is that their performance falls, but on daily subsequent sessions, they will begin to learn that they will begin to report to you the presence of the light stimulus. And if you move the light so these are data from 5 different animals over time, and you can, over sessions, with a brief pulse of light over the glomerulus. If you move the light off the glomerulus, or you use control animals that don't express channel rhodopsin, the animals are not able to do this task. So for the first time, we can now address a specific glomerulus in a behaving animal. And we did a number of experiments. So first of all, this question, we're able to show that, in fact, animals can report to you the activation of a single glomerulus. And this is very different than an odor stimulation because we're activating one glomerulus and not both. But even under those conditions, the animals can do it. So what I'm diagramming here is another experiment that we did. What we can do, here's a diagram of the glomerular array. Our channel rhodopsin expressing glomerulus is surrounded here by yellow. And we have 2 different conditions. One condition is in the absence of light, and one is the presence of light. And we can train the animals, as I just showed you, to discriminate between these 2 conditions. And so activation of a single glomerulus, the animals will report that to you. If you do a similar experiment and you also present them with an odorant, the animals can also distinguish these 2 conditions. So what I'm diagramming here is that this odorant carvone activates a number of glomeruli. It does not activate the native M72 glomerulus. And if you supplement this response pattern with a new glomerulus, the animals will report that to you as well, showing that even on the background of a natural odor, addition of a glomerulus to this repertoire of glomeruli will change, presumably, the perception of that odor. The animals can report that to you. If you use an odorant that activates M72 natively, the animals don't do that task very well at all, suggesting that the odor can actually block or mask the input from the light. All right, so we can show that animals can detect the presence of activity in a single input channel in the presence of odor or in the absence of odor. In addition, one surprising thing is that the animals will also report to you that light intensity through a single glomerulus, that is to say the intensity of input through a single olfactory input channel, can be perceived by the animals. This is an experiment that you can't do with odors, because if you increase the concentration of an odorant, you also recruit new glomeruli. So these data suggested that for the first time that glomeruli are not just binary inputs, they're either on or off. In fact, the input, graded input in an olfactory glomerulus can be read out downstream by the system. Right? So we showed that activation of a single glomerulus is sufficient to drive the behavior. You can do this in the presence or absence of odor. And a single glomerulus can encode stimulus intensity and also timing. I haven't showed you anything about timing. But the idea, the conclusions from this work, is that each individual input channel is actually multiplexing information on a number of different things. Right? So we have both the identity of an odor, the timing of when the activity comes in, and the intensity signal. Okay? So this made us think, well, okay, maybe the individual lowly glomerulus is actually contributing significantly. If we were to go in and remove glomeruli, remove genes, we might be able to have some hope at seeing changes in behavior. Okay, so how do we do this? So the approach that we're taking is to exploit the one-to-one nature between odorant receptor genes and the neurons that they express. If you remove an odorant receptor gene from the genome, the receptor is no longer there. What happens is that the glomerulus essentially goes away. Right? You don't have a glomerulus that's corresponding to that particular receptor anymore. But what receptor are you going to delete? There's a lot of them, and it's very difficult without a priori knowledge of what they respond to, which ones that you might want to go after. And so I lied to you when I showed you this graph. In fact, the graph looks more like this, right? So there is a small family of receptors which were identified in 2006 by Linda Buck's lab, by Steve Lieberlus, who's now at Harvard. And these are the trace amine associated receptors. And so the true phylogenetic tree of the olfactory receptor repertoire of the mouse looks something like this. So we have about 1,000 classical ORs, and we have 14 olfactory tar genes. And this was discovered at about the time that my lab started. And we looked at this and we said, well, this looks like a wonderful opportunity to begin this approach of getting rid of receptors in a controlled way and looking at effects on perception, because we figured that a family of genes that look like this, they're all clustered, they're similar to each other, you're most likely to see effects on behavior by manipulating this gene family. Okay, but not much was known about the TARs. In fact, our view at the time of the system was something like this. What I had shown in my postdoc and also going into my own lab, we finally published this in 2009, was that these two classes of odorant receptors, which are imaginatively named class 1 and class 2, they are projected onto the surface of the olfactory bulb. So neurons that express the class 1 genes tend to project to glomeruli in this dorsomedial region of the olfactory bulb, and the class 2 genes, those neurons project to the lateral and ventral olfactory bulb. It's essentially the rest of the system. When the TARs came out, it was entirely unclear how they fit into this setup, this model. What we did know was that, at least with respect to the class 1 and class 2 genes, is that we could see that certain odorants, certain classes of chemicals, preferentially activated one region or the other. The regions are named D1 and D2 for dorsal 1 and dorsal 2. And in this image it's shown in grayscale. The idea is here, this is the D1 region. If you put on short-chain organic acids, which tend to smell awful, they activate preferentially glomeruli in the D1 domain, by and large, although not always exclusively. Certain ketones will give you preferential activation in the D2 domain, and but a lot of odorants activate both. And so there was some idea that maybe there's some topography relating to chemical structure here, but it wasn't entirely clear. So the general idea is that we have a bunch of sensory neurons intermingled in the epithelium, and what we have evidence for is that a subset of these receptors, these sensory neurons, look to the class 1 gene repertoire and preferentially choose from those receptors. And we have a subset of neurons that preferentially choose from among the class 2 genes. And in data that using data that I don't have time to cover, what we've shown is that these neurons, their axons segregate from each other in the olfactory nerve and project onto the olfactory bulb. So that nice order that I showed you in the projections from the nose to the bulb seems to be conferred by the nasal epithelium itself. So the neurons themselves seem to contribute significantly to the organization of the bulb. All right, that's an advertisement for the olfactory epithelium, but really what I want you to appreciate is this organization. And when the TARs came to be, we didn't know how they fit in. We did know, through work from my graduate student, that a majority of the TARs, 10 out of the 14, are expressed in this region of the epithelium, and we have to account for them somehow. In addition, Steve Lieberless had shown that if you express some of the TARs in vitro, they respond to volatile amines. So let me call your attention to the very nice gene structure here, which is that the TARs are all found, the TAR genes are all found in a single cluster in chromosome 10 in the mouse. The first member of the family is the canonical member. It's actually not expressed in the olfactory system. It's expressed in the brain. It responds to trace amines such as phenylethylamine and tyramine, also to amphetamine and MDMA. And there's a whole group of people working on this receptor. The rest of the genes, for many years, had no known function, but now they're known to be expressed in the olfactory epithelium. So what Steve did is he expressed some of these in heterologous cells, in HEC cells, and he was able to show that some of them, for example, here's TAR4, which I'm going to talk about at length, is activated by phenylethylamine, but not the other amines that he tested. Okay, the concentrations that are required to activate these receptors tend to be in the micromolar range in these assays, so keep that in mind. So that's about all we knew. You could put them in vitro and they respond to volatile amines. That sounds wonderful. We didn't know anything about where the glomeruli for the TARs are located in the olfactory bulb. So we used our genetic strategy. We could take the TAR4 gene, which is located here. We could insert a red marker after the gene and label the cells that express the native TAR4 locus. And what you can see is that you get a population of neurons in the dorsal epithelium that have chosen the TAR4 gene to express. They turn themselves red. More importantly, if you look in the olfactory bulb, you can see that the axons of these neurons project to glomeruli in this, again, this dorsomedial region. So the TARs are projecting to the main olfactory bulb. That was news, perhaps not surprising. But the next thing that we went on to show was that if you target two of the TARs together, TAR3 and TAR4, you can see that their glomeruli fall out very close to each other, suggesting that perhaps there's a region of the olfactory bulb that's dedicated to the TAR glomeruli. So what we wanted to know was how the TARs fit in with our two known D1 and D2 projections. Again, without going into the details of the experiments, you can do a little genetic trick, which is to replace the coding sequence of one of the TAR genes. Reasons that I get into, what that allows you to do is to visualize the other TAR glomeruli. And if you do that manipulation, what you see is that, in fact, a majority of the TAR glomeruli are located in this region of the olfactory bulb. We did this with tar4. We also did it in another strain with tar9. It doesn't label as well for a number of reasons, but it allows you to make a picture like this. So you can now see that there seems to be a little pocket of glomeruli dedicated to these tars, at least a subset of them. So after several years of work in my lab, we finally came up with a revised model for the system, which is that these neurons are intermingled at the epithelium, and in fact there is a small compartment that represents the tar genes. So there are all sorts of questions here. Why? Why is the system parsing these different classes of receptors, of odorant receptors or olfactory receptors into different regions of the olfactory bulb? Short answer is, we don't know. That's one of the things that we want to understand. The other thing is, well, how do the tars actually contribute to olfaction? The olfactory system has lots of receptors. There's over 1,000 odorant receptor genes. What is so special about these receptors? Why are they required in order for the animal to do whatever it has to do? So I mentioned before that Steve had shown that some of these receptors respond to volatile amines, but this was all in vitro work, and we wanted to see how the neurons that express these receptors behave. So what we did was we took our gene-targeted animals, in this case for tar4, and we performed electrophysiological experiments in which we use perforated patch recording onto the dendritic knobs of these sensory neurons. And so this is very close to where the transduction mechanism is in the cilia of these cells. This is in an explant of the olfactory epithelium, which is sitting in a dish, very similar to a slice, an acute slice. And we can come in, we can puff on odorants in the aqueous phase onto the cilia of these semi-intact cells, and we can record transduction currents from the knobs. And this is a very sensitive measure for whether or not you've activated the receptor expressed by that set of neurons. And when you do that, this is the response to phenylethylamine. What you get is you put on the stimulus, you get a downward deflection in the trace, which corresponds to an inward current in the cell. You can also see that the cell is happily spiking away because we don't have very good space clamp by the nature of the kind of recording that we're doing. But you can you have a very sensitive measure of transduction. And what we immediately found was, well, this isn't so surprising. In vitro, tar4 responds to phenylethylamine. However, it doesn't respond to anything else. In our assay, if you look at other amines, what you find is that those cells also seem to respond to other amines as well. They're just not as sensitive. The receptor is just not as sensitive. If you genetically remove the receptor or you swap it for another receptor, all of these responses go away, suggesting that all of these responses are mediated by tar4. All right? So tar4 ligand, very similar to other receptors, but in fact it's also broadly tuned. If you look at the profile of these cells at 10 nanomolar, which is actually for odorants a very low concentration. Usually people are using micromolar concentrations. You get a very broad response profile to a number of different amines. If you lower the concentration to half nanomolar, you can see now the best ligand here is phenylethylamine, but you also get responses, small responses to the others. We did this also for tar3, and this becomes